KPV is a tripeptide made of lysine, proline, and valine. So K is lysine, proline is P, and valine is V. And it's a tripeptide, so there's just 3 amino acids, and they're all linked together. And you'll often see it called alpha-MSH 11 through 13. And there's a reason for that. So alpha-MSH is alpha-melanocyte stimulating hormone. KPV is its C-terminal 3 amino acid tail at this position 11 through 13. And this origin here is pretty important because alpha-MSH, it's part of the melanocortin system. And this system touches on things like pigmentation, on inflammation, on immune signaling, as well as tissue protection. So let's look at the differences between the two. So alpha-MSH, it can signal through melanocortin receptors. These are something called G protein coupled receptors. And basically they're these larger receptors where a peptide or molecule can come and dock onto this receptor, and then it's going to cause this big cascade inside the molecule. So for the case of alpha-MSH, you're going to have it come and dock onto, say, one of the melanocortin receptors. Say it's melanocortin 1 receptor, and then the melanocortin 1 receptor is going to change its shape based off how alpha-MSH docked, and then there's going to be a whole bunch of changes inside the cell. I promise this is important for our KPV discussion here. So with alpha-MSH, it has kind of these broad anti-inflammatory effects, but one reason KPV drew attention is that alpha-MSH, it's anti-inflammatory, but it also drives pigment production. Whereas reviews describe KPV as kind of keeping this anti-inflammatory side while dropping the pigment-inducing side. Real quick, if you're finding this helpful, please check out the peptide database at join.bioavailable.app. It's completely free, have no plans to ever make it paid, just to help you out. Thanks. This is the melanocortin system, okay? It starts with this precursor protein you can see in the top left called POMC, and that's cut into hormones, including alpha-MSH and ACTH. ACTH is adrenocorticotropic hormone. And alpha-MSH, though, is this 13 amino acid peptide, as we mentioned. And the part here, if we're looking at this molecule, that kind of pink, white, and blue molecule, this is alpha-MSH. And the part that switches receptors on is this 4 amino acid core in the middle. The histidine, phenylalanine, arginine, and tryptamine, or HFRW component of the molecule. So KPV is at the other end, at the C-terminal tail here. So the kind of active site or the binding motif is what you call it in biochemistry, is the HFRW component of alpha-MSH. So KPV is not exactly binding in the same way that alpha-MSH would. That's all that I'm trying to say here. And just a quick note at the bottom, all these G protein coupled receptors or GPCRs, they all signal CAMP or cyclic AMP, but they do it in different ways, so they can all have different effects, and that might be helpful for later. And one quick thing is that I have a video a bit more on the melanocortin system. So if you hop over to my melanotan 1 and 2 video, I go into this in a bit more depth. But for the sake of this video, there are 5 melanocortin receptors. There's 1 through 5. And again, I mentioned they act on tissues from, the skin to the immune cells to the brain, and these are all G protein coupled receptors, and they signal by raising this cyclic AMP inside the cell. Okay, so here's how alpha-MSH normally signals. So that HFRW core sitting in the middle of the peptide is the part that actually docks onto a melanocortin receptor on the cell surface, and that kind of binding event is what switches on the receptor in the first place, and it sets everything else in motion. So once that receptor is activated, it engages this GS protein on the intracellular side, so now we're inside the cell, and it in turn switches on adenylyl cyclase. And this enzyme raises cyclic AMP inside the cell, and then rising cyclic AMP activates PKA. So this is the kind of the core signaling chain, and from here, the path can kind of fork depending on what kind of cell you're in. So in pigment cells specifically, PKA goes on to drive something called MITF, and this then leads to melanin production. And that's the visible color effect that you get from, tanning or from, say, melanotan 1 or melanotan 2. You get that melanocortin 1 receptor activation leading to an increase in pigment. So that's just one downstream branch, and that's just used as a concrete example here of where this kind of signal can lead with alpha-MSH. But the same cyclic AMP signal, it also branches a different way in immune cells and in barrier cells. So there it helps block something called NF-kappa B, and NF-kappa B is another signaling molecule or inflammatory kind of upstream molecule, and it helps block NF-kappa B activity, and it keeps this other molecule, we can just call it IKB, it keeps it stable rather than letting it get degraded. And the practical result here is a drop in inflammatory output, where we're getting less of these inflammatory cytokines. So these inflammatory molecules that basically just give us tons of inflammation, namely TNF, interleukins, as well as nitric oxide. So the kind of net effect of blocking NF-kappa B is we get less inflammation. And so this kind of anti-inflammatory branch here is the part most relevant to KPV, since it's this branch of the pathway that KPV manages to engage even without the receptor binding core. So the receptor binding core being that HFRW site on alpha-MSH. So it doesn't have that, it's just that KPV tail. And this picture here, it really comes down to one contrast, and I keep on mentioning it, but I'm going to continue to do so because I think it's helpful. Full alpha-MSH, it has the HFRW core sitting right in the middle of its sequence, and that core is what lets it dock onto melanocortin receptors. Once it does, it raises cyclic AMP and triggers both of its classic effects. So we see things like pigmentation as well as the anti-inflammatory response. Now, KPV is missing all of that. It's just the C-terminal tail with no HFRW core to speak of here. So that doesn't exist. So it barely nudges cyclic AMP at all. But it doesn't quite stop there, because losing the core here, it doesn't mean that you're necessarily losing all of the activity. KPV still keeps the anti-inflammatory effect, it just sheds the pigmentation side that normally comes bundled with it. And because KPV is so small, it's not limited to working through a receptor on the cell surface. So theoretically, it could actually be carried directly into cells, say in the gut, by hitching a ride on something called the PEP-T1 transporter, the PEP-T1 transporter. So here's that mechanism illustrated in a bit more detail. Okay, so KPV, it's just the tail end of alpha-MSH. It's missing the HFRW core. And so how is it doing anything at all on its own? And so there's two competing explanations for this. I'll show you this first one on the left in pink. And it says that KPV is still somehow finding its way onto a melanocortin receptor, and it's binding somehow just weakly or incompletely. So just that little tail piece is somehow maybe able to potentially activate it partially. But the problem is that the data don't really back up this idea in its classic form, because if KPV were activating the receptor the normal way, you'd expect to see a rise in cyclic AMP, since that's kind of the textbook signature of melanocortin receptor signaling. So that rise just isn't there, which makes a true receptor-driven mechanism here hard to defend. The second explanation here in green on the right, it fits the evidence much better. So instead of working from the outside via a receptor, remember when we signal a G protein coupled receptor as you see on the left, it has to be done from outside the cell. So it doesn't actually, the molecule, the signaling molecule, does not travel through the cell. It just changes the shape of the receptor on the outside. So with though something like PEP-T1, so with PEP-T1, KPV could actually theoretically get pulled into the cell. So in the gut, it could hitch a ride through PEP-T1, and this is the transporter that normally moves small peptides across the intestinal lining. And once it's inside, KPV could work from within. So it keeps something called P65 from migrating into the nucleus of a cell, and it helps stabilize IKB so it doesn't get degraded, and then therefore dampens this interleukin-1 beta signaling, or IL-1B. So I'll explain this a bit more. So let's talk about the P65 bit. So here's a close-up of this kind of different door. So we're thinking about PEP-T1 at this point. So KPV, it's gone through PEP-T1 and it is inside the cell now. So we have NF-kappa B and P65. So the inflammatory switch, NF-kappa B's P65, it normally gets carried into the nucleus by a shuttle protein called importin alpha-3, where it then switches on these inflammatory genes. And a 2012 study, it actually linked KPV to less P65 import into keeping this IKB stable. So it basically holds P65 out in the cytoplasm or outside of the nucleus. And then the net effect is fewer inflammatory genes that get turned on. And so the molecular version of the alarm switch is not kind of able to get in to the nucleus to have an effect on the genes. So we can see here that at this importin A3 getting into the nucleus phase, we can see that KPV is blocking that part right there. Because in order to kind of have genetic modification or epigenetic modification, we would think that it's important to be able to have some nuclear signaling that would change the DNA. And by nuclear signaling, I just mean maybe there's some change that causes the DNA inside the nucleus to, alter its structure somehow. And then you could have changes like fewer inflammatory genes that are activated. Okay, so let's look at some of the studies now and get a little bit into some of the more animal and preclinical data. And again, when I say preclinical, I just mean animals in cells in any kind of gene lines, just not in humans, because there are no human studies that look at KPV. So a 2003 pharmacology paper here, it looked at mouse peritonitis, and it's this kind of animal model where inflammatory cells, they flood into the abdominal cavity. And the researchers here, they gave KPV systemically. So systemically typically means they probably injected it. And then they counted how many inflammatory cells showed up. And what they found is that KPV reduced that inflammatory cell accumulation. And this is a clear, concrete anti-inflammatory effect in a living animal. So it's not a complete human claim, but it is a bit of a signal here that's helpful. And then came some mechanistic surprises here. So the effect was not blocked by a melanocortin 3 or 4 receptor antagonist. So we remember those melanocortin receptors. So it still worked in mice with non-functional melanocortin 1 receptors. So we're starting to think here, how can something work if our melanocortin receptors are not working? So is this because maybe it's getting into the cell through a different way? Or sorry, I'd like to correct myself there. It is getting into the cell in the first place, and maybe it's not even activating the melanocortin receptors. And in macrophages, KPV did not raise cyclic AMP the way classic melanocortin signaling would. So these authors argued KPV more likely acts by interfering with interleukin-1 beta, which is an inflammatory signal, rather than by flipping a pigment receptor. Human keratinocyte work here, it adds a little bit of a nuance. So keratinocytes are the human skin cells. And in those skin cells, KPV and related peptides did not raise cyclic AMP either, though calcium signaling here appeared under certain conditions. So even in skin, the signaling here is not necessarily just one clean switch. And so the first big takeaway is that KPV is related to alpha-MSH, but it has its own signaling logic here. So if somebody just explains it as it's a melanocortin receptor drug, that could be partially true, but we're not entirely sure. It might be more likely that it maybe works through PEP-T1 as an example. So now if we zoom out to inflammation itself, so let's think a little bit broader. Inflammation here is not just swelling or pain. It's actually something you could think of as like a traffic system. So injured tissue, it sends out these kind of cytokine flares, and cytokines are just these signaling inflammatory molecules. And the injured tissue also sends out, immune cells, things like neutrophils that move toward the signal. And so KPV's earliest kind of pharmacology is partly about this traffic here. So in the peritonitis model, fewer inflammatory cells showed up after KPV. So one of its effects is simply turning down the volume on cell recruitment here. So we're getting less cell recruitment to that inflammatory site. And there's another thing that happens here in epithelial cells. So these epithelial cells are the lining cells of the gut, the airway, and the skin, and they have triggers like TNF-alpha and interleukin-1 beta that push the cell into an alarm state. So they tell the cell, sound the alarm, and they tell it to kind of crank out inflammatory mediators or just inflammatory products again. And one of the main alarm pathways is something called NF-kappa B, or we can think of it as NF-kB, okay? You can see it there. Picture it as an alarm switch. It's kind of waiting just outside the nucleus. And when NF-kappa B's P65 component gets carried into the nucleus, the cell starts switching on inflammatory genes. So this is what we talked about earlier with the theoretical mechanism of KPV and how once it gets inside the cell, it has an effect on NF-kappa B and P65 translocation or movement into the nucleus. This is all what I'm talking about right here. In a 2012 study, it was on bronchial epithelial cells. It connected KPV to exactly this step. So quickly, I'll just tell you, in our body, we have a bronchus. You can just think we have our trachea, and then it deviates into bronchi. So just think your airways. So it's your airway epithelium. So the cells lining your airways, okay? And that's what this study was. So KPV was linked here in this study to less nuclear import of P65, as well as to stabilizing IKB, and this is the protein that holds NF-kappa B outside. So it stabilized the very thing that kept, P65 or NF-kappa B outside of these cells. So in plain terms, KPV basically makes it harder for this P65 alarm switch to get carried into the control room here and flipped on. So that's the best kind of analogy I can give for this kind of situation. And that same 2012 study, it was done in these bronchial epithelial cells, which I mentioned. And again here, it tied KPV to reduced P65 import and stable IKB, giving this specific mechanism, not necessarily just generic lowering inflammation. It tied that lower inflammation to a specific mechanism of KPV. And when the pathway was dampened, then the downstream outputs fell too. So inflammatory mediators like interleukin-8, eotaxin, and MMP9 activity, they all came down in the model. So these are all inflammatory markers that went down as a result of KPV administration, and likely they were downstream from the less P65 import because we had some certain inflammatory genes that got turned down. And some gut studies also added a second pathway. So KPV also reduced MAPK activation. It's another route cells use to respond to stress and inflammation, and it matters in two cell types. So both epithelial cells as well as immune cells. And that's why KPV is interesting for barrier issues here, not just for immune cells. So immune cells you can think of commonly as being extremely important in our inflammatory cascades and pathways, but they can also be problematic, as well as we can have problems with some of these signaling molecules if it's in excess, and we can have this chronic inflammation that can cause a whole array of different problems. But the same can be said for epithelial cells, which start sending off all these really large, chronic inflammatory signals for a long time, and you're going to have a whole bunch of problems as a result. So KPV has effects on both epithelial cells as well as immune cells. That's all I'm trying to say. There's a catch though. Anti-inflammatory does not mean that something's automatically good. So inflammation also fights infection and it starts repair. So we just want to keep that in mind, that just dampening an inflammatory cascade, it could also have some downstream effects that might not totally be always a good thing. And so, yeah, that's all I'm trying to say there. The gut is KPV's strongest lane here because it connects the molecule to a transporter as well as to some disease models. So the key gate here is something called pepti-1. It's a transporter that carries di, or 2 amino acid couplings there, or 3 amino acid peptides across the gut lining. So this is exactly the kind of door a tripeptide like KPV can use here. And normally pepti-1 lives in the small intestine, but the crucial point here is that pepti-1 can be induced in the colon during inflammatory bowel disease. So the inflamed tissue itself opens a door KPV can enter through. And the central paper here is a 2008 gastroenterology study. Gastroenterology study is just the gut. You can think of it as the gut. And it showed that KPV is taken up through pepti-1 in intestinal epithelial cells and immune cell models, and then tested oral KPV in DSS and TNBS mouse colitis. These are just 2 standard experimental colitis models. So we're basically have these 2 types of mice that have different gut problems that are related to inflammatory bowel disease in humans, and they're kind of trying to replicate that model in the mice. And so their findings were that at nanomolar levels, KPV reduced NF-kappa B and MAPK activation, and it also lowered pro-inflammatory cytokine secretion. And the oral KPV reduced inflammatory markers in the mice measured by histology. This is a marker. This is something that's used in the experiments, as well as cytokine mRNA. So this is one of the clearest mechanisms to affect links in the whole KPV literature here. So this was a very interesting study, and it linked it to pepti-1, as well as all these downregulation of inflammatory markers in these inflammatory bowel disease mouse models. There is a limit, though, here is that it's just not human. So it's not a human study. It was in mice, but there is some promise here in the preclinical literature. And a second 2008 paper tested KPV across 2 intestinal inflammation models at once. So they had one that was DSS colitis in the mouse, and another that was this other type of transfer colitis, where they kind of give a mouse a problem. So they take one mouse and they take its problems and they give it to another mouse and give it inflammatory bowel disease. That's the best way to explain it here. And KPV treated animals recovered earlier. They regained more body weight. They had fewer inflammatory infiltrates, and they had lower myeloperoxidase activity in the colon. So less myeloperoxidase just means basically you can think of it as less inflammation. And in melanocortin 1 receptor defective mice, KPV rescued every treated animal from death during DSS colitis, which is just more evidence that the gut is at least partly independent of melanocortin 1 receptor signaling, and kind of weakens that initial mechanism I showed you of how KPV doesn't necessarily work the same way as alpha-MSH. In a 2016 colitis-associated cancer model, they added a clean kind of check here. So KPV's effect on tumor development disappeared in pepti-1 knockout mice. And so that does not prove, though, cancer prevention in people, but it does pin pepti-1 as the gate that the effect runs through here. So the gut takeaway is quite balanced, and this is the strongest preclinical KPV lane in that in the GI models here, there is good evidence that KPV could be somewhat anti-inflammatory. So there is questions, could it be helpful with human ulcerative colitis or human Crohn's disease? But again, this is just a question, and it hasn't been shown in humans, so we don't have any clinical trials for that right now. Now, there's also a delivery problem too, and for KPV, this is quite important. A peptide swallowed as a free drug faces, your stomach acid, so really low pH. It's going to face a whole bunch of enzymes, mucus, and part of the question here is where it's actually released in what kind of effects you're going to have, because if you release it in the stomach, it might be way lower of a pH than maybe if you released it in the colon, for example. So one colitis paper actually notes that plain KPV solution is very unstable if given rectally, which compromised its effect. So these colitis papers, typically the administration, say it's an ulcerative colitis mouse model, it might be given rectally because that's just an easier way to access the sigmoid colon, which is just the lower part of your colon, to be able to help with the problem. So KPV is unstable. And PEP-T1, it does make uptake plausible, but it doesn't solve the exposure. So I want you to think of it like mailing a fragile package. So there's this mail slot that's at the destination that doesn't really help if the package falls apart in transit or lands at the wrong address. So the peptide still has to survive and it has to reach the inflamed site. And so release there has to be there at a meaningful dose at the site. So the delivery system is what protects the package and kind of drops it in the right place. So we have this kind of delivery problem as well. Back to some of the studies though, a 2010 gastroenterology paper did exactly that with this delivery issue. So it built these nanoparticles carrying KPV, and then it wrapped them in this hydrogel. It was this alginate chitosan hydrogel, and it was designed to target the colon, and it was tested in the DSS mice. And so that's very different from just swallowing free KPV and, hoping that it would make it to the gut in the mouse model. And so the headline result was efficiency. So this nanoparticle system reached similar effectiveness at a KPV concentration reported as 12,000-fold lower than a free solution. So the vehicle here, and the vehicle is just the method of administration, was doing a lot of work here. So it actually was really helping the KPV get to the system. And so in 2021, an ulcerative colitis model used this self-cross-linked polyglutamic acid gel. So it used this stable gel to stabilize KPV and slow its release for better rectal delivery. And a 2022 double network hydrogel was designed to stick to inflamed colon mucosa. And so the mucosa is just the kind of epithelial lining of the gut. So it's designed to stick to inflamed colon mucosa rather than healthy tissue. And in rats, it improved mucosal barrier recovery and colitis measures. So we have this lesson here where when we have smarter delivery, we can have a better local effect if we pair it with, say, a hydrogel, which is a really common example of trying to improve a delivery system. And the newest entry here is a 2026 Science Advances paper using a self-immolative prodrug conjugate that's called pro-KPV. And so in plain terms, this one specifically, it's just engineered for oral stability, as well as for mucus penetration and reactive oxygen-triggered release right at the inflamed sites. So a micelle is basically this lipid or fat nanocarrier. So basically you can have the KPV is all kind of clumped together, and then you have this fat, these kind of phospholipids that surround the KPV core. And because fatty acids are charged, they have kind of a polar end and they have a non-polar end. This can be, not fatty acids, but phospholipid bilayer. You can have this polar end and non-polar end. And so in colitis mice, pro-KPV reached 3.8-fold greater colonic accumulation than free KPV and worked at a 20-fold lower dose. So the same platform also accumulated in inflamed lungs and reduced injury in an acute lung injury mouse model. So this is interesting as well as current, but it's still an engineered preclinical platform here. So here's a practical take-home point here. So the delivery tech, though, is not the product. And so it's something you could just think of as, what's the vehicle? How are we delivering this product? Does the receptor activation actually work how it's described in the literature? There's also some wound and skin studies, although it's not identical to the results that were found in the gut. The cleanest animal result here is a 2006 rabbit corneal study. So the cornea just goes over your eye. It's a covering. And researchers scraped off the corneal epithelium, and then they applied KPV topically to the healing eye. And at 60 hours, all 8 KPV-treated corneas were fully re-epithelialized, and so basically it healed back. And so were all 8 treated with substance P as a positive control, while none of the placebo corneas had healed. And when they pre-treated with something called LNAME, which is a nitric oxide blocker, the KPV effect disappeared. So all this is trying to say is that nitric oxide was part of how it worked in this model here. In diabetic mouse wounds, a 2022 film dressing layered KPV and epidermal growth factor. KPV released early and EGF later, and it improved repair markers. So when KPV is combined with the growth factor, can we really hand all the credit to KPV here, or is the growth factor more responsible for the kind of repair that we've seen? In rat chemotherapy-induced oral mucositis, a 2021 mucoadhesive, so that's just something that can attach to our mucous membranes, or say your below your tongue, for example. This 2021 mucoadhesive KPV hydrogel improved food intake, body weight recovery, tissue repair, and inflammatory cytokine measures. Again, this is just a specific delivery system, so it wasn't free KPV. And a 2025 keratinocyte study looked at fine particle pollution, so PM10, in human HACAT cells in a 3D skin model. And KPV reduced the reactive oxygen species linked damage, including interleukin-1 beta and NF-kappa B, as well as MAPK-related effects. So we have the same anti-inflammatory fingerprints here, and they're now in skin cells. A 2019 review framed melanocortin peptides like KPV, or similar peptides to alpha-MSH like KPV, as future candidates for wound and ulcer treatments. And they were candidates though, so not exactly that they were established treatments. Now, we're missing human evidence. So this is a huge gap in the literature. We don't have much data on humans. In fact, we have 0 data on humans. I wasn't able to find any clinical trials in humans, so please drop it in the comments if you know of any randomized control trials that were done with humans, even retrospective studies with humans, but I wasn't able to find anything. So yeah, please let me know. And on July 23rd here, so in about a month's time, we're going to have some information come about KPV because there's the FDA PCAC meeting. And what this is going to be discussing is there's going to be 2 compounds. There's going to be the acetate version of KPV, and there's also going to be the free base version of KPV. But you can think of it, they're both KPV. And this is to talk and discuss, these FDA committee members are meeting to discuss if KPV should be reclassified and be able to be compounded in American pharmacies. So when something's compounded, a physician could theoretically prescribe a compound off-label to treat another condition. And so that's what this kind of whole meeting is there to discuss. There's a few other peptides that are going to be discussed at the meeting, but this will be kind of decisive as to whether in the future these could theoretically be compounded. KPV is a tiny alpha-MSH derived anti-inflammatory signal, and it's got some very interesting preclinical gut, barrier as well as delivery evidence. But it's also not proven as a human therapy, and so I think it would be great if some labs started to do some clinical trials on this. But again, it's always tricky to get clinical trials done in some of these naturally occurring molecules. So with that, thanks so much for watching. If you have any questions, please drop them in the comments, and hope you all have a great day. Bye-bye now.